So today's webinar, it's titled A Guide to UniProt for Students, but really it's aimed at anyone who is relatively new to using our resource. So I'm Paul Denny, and helping me today is Dave Carpentier. We're both part of the UniProt content team, and our job is as curators to add up-to-date information and update the information that's there already on the UniProt website for proteins. And the website parcels up the data in a number of different ways. We have a knowledge base, which essentially consists of a mixture of automatically annotated sequences, so the unreviewed part of the website. So these are automatic annotations created in the process of adding them to the database. And then the reviewed section of the collection of proteins that make up the knowledge base. These have been manually curated, as well as previously they would have been through the automatic annotation pipelines. So that's one way of thinking about the data in UniProt. We also divide up the proteins by their species of origin. So all of the known proteins from a whole genome for a species, and we call this a proteome. And then there are things called protein clusters. These are collections of proteins with different levels of sequence identity. So we'll cluster them together based on several thresholds, 50, 90, or 100%. And then finally, there's an archive, which actually is the starting point for all these other databases, UniProt. And this is a non-redundant set of protein sequences, which will include things which haven't yet been through the automatic annotation pipeline, and they will ultimately go through that pipeline and then join the unreviewed section, some of them, in UniProt. So what I'm going to do is I'm going to, with Dave's help, show you how we can search for proteins, find out data about them, functional information about those proteins, and then go from the functional data to sequences for those proteins and things like post-translational modifications. And then say just briefly a little bit about proteomes, and then mutations in those proteins and how they have a role in disease. So what is UniProt? It's a collection of high quality, freely accessible protein sequence and function information, including the sequences, as I've said, physiological information. We focus on the normal function of proteins. And this will include things such as subcellular localization, any pathways of proteins involved in reactions or interactions of those proteins, and of course, as I've mentioned, involvement in disease. On top of that, there's structural information, including the topology of a protein. For example, if it's in a membrane, where are the N and C termini and membrane spanning parts of a protein, and also give access to 3D structures. And this includes both experimentally derived structures and predicted structures from tools like AlphaFold. And then together with those data, we offer various tools such as BLAST and multiple sequence alignment. And all of this is put together by three big partners, EMBL-EBI at Cambridge, Hingston, Cambridge, the PIR in Washington, DC and Delaware in the States, and the Swiss Institute of Bioinformatics, Geneva in Switzerland, we're the three major partners in this consortium. This slide kind of describes what the website consists of and the principles of using it. You can download information from it. You can visualize protein information in different ways. There are different ways to search, simple searches, more advanced searches. And then there are also ways to put in a whole batch of identifiers or retrieve more than one sequence at once. The way the data is then presented to you can be customized. You can change the view, the exact information that's portrayed, and within each entry, and I'll take you through one of these in a minute, there is a whole wealth of data, including provenance, that we're very keen on being able to say, where does this data come from? What's the evidence? And as I've said, there are various analysis tools, and I'll say a little bit about those later. Where does the data come from, which makes up what's in UniProt? So we've got three main sources. The biggest one is the International Nucleotide Sequence Consortium, so the ENA here at the EBI in Hingston, the Japanese equivalent, DDBJ, and then CBI in the States. And these nucleotide databases, protein sequences derived from those are the major source of data that comes into UniProt. But we also pull in data from Ensemble, the web browser, again here at the EBI, and from PDB, the structure database. And then all those data are integrated. They go through various analyses and then organized by the curators. This is our homepage. And if we start at the top left corner here in green, there's a little box outlining the various tools associated with using the data on our website. Top right in blue, little blue box, this shows you the most recent update of the data. These get changed approximately every 8 weeks. So this release number reflects the most recent data set, and there are statistics that summarize that. So it's this one, 2024-04 is the current release. And I've mentioned a little bit about the different ways of viewing the data here across the bottom in blue. Going to the search bar in the middle of that last slide. In green here, where it says select database, we talk about the knowledge base UniProtKB, but you can also reduce the search that you make from the knowledge base to a particular set of proteins, so things involved in disease. And in this search bar in the middle, you can type anything in, any text search, a gene name, a disease name, and so on. And then you'll notice over here in purple, where it says advanced, quite small text, but nice big friendly text at the top there. This is a way of doing a more sophisticated search, which I'll say a little bit about in a minute. And finally, this thing called list. So let's say you've got a list of identifiers from either from UniProt or from another database. You can use this if you click on this link here where it says list, and you can put in a list of IDs and then do a search. And I'll show you an example of that in a minute. So in this example, I've chosen a set of proteins that are involved, in this case, in the process of autophagy, or autophagy, as some people would prefer to say it. And these are the gene names. So ATG12, for example, is a particular gene name. And we use gene names within UniProt as a kind of, as close as possible, universal identifier for each protein. So there'll be some variations in that, but mainly we use gene names. But of course, that starts getting more difficult if one's dealing with a non-human species. But I won't go into that now. So you can search for gene names either one by one in the search bar, as we just saw a moment ago, or you can do a more advanced search. So this is an example of an advanced search where you can select which particular database you're going to search in. You can choose a whole range of different fields. So that in orange here is the taxonomy box. So we could choose to say, well, we only want to look at human or mouse or fruit fly. then there are different ways to link together using Boolean terms and or not. You can link together multiple keywords or other different, you can choose other fields. In this example, we've got a keyword, but we could select from the drop-down menu and choose a different field of data which is stored in UniProt. And then you can add yet more by clicking on the Add Field box. I mentioned earlier ID mapping. Well, that's the same thing as this little list here. So it allows you to take a list of proteins or a list of gene names. In this case, I've got 3 gene names in this example, ATG5, 12, and 16. And if I now choose only human to restrict the organism, and I've told the website on this particular dialog box that I'm choosing from a list of gene names. I've got a list of gene names here in green. And I want to get UniProt accession numbers back. So if I was to do this, I'd get something like this. And this is a summary of the results. This doesn't actually show you the full results. I'll show you that in a moment. This is a toolbox. And what this does is it holds on to searches or analyses that you've done for 7 days. If you want, you can keep them for up to 14 days. So you can carry on coming back to your results and see what they are if you want to keep a record of them on our website. But after 14 days, they get deleted. So this is what the top of the search bar, the top of the web page looks like. This is the little icon that indicates the toolbox. And if I now show you what the results would look like if I opened up the results from that previous window, in the test that I did in order to create this image, I accidentally put in a gene name, or it's a gene name, which doesn't exist. And this one here, ATG161, wasn't mapped to anything, unsurprisingly, because it's not used for any gene. And in the left-hand side here of the window, you'd see ways of different filters. So you can filter which results you're interested in. So let's say you're only interested in any proteins which have 3D structures. So there are 2 of them. Or you're only interested in entries which have been reviewed by a curator. Again, there's 2. At the beginning, I mentioned a little bit about reviewed versus unreviewed. So just to expand on that a little bit, we get as curators, we get almost all our information that we add to a particular protein entry from the literature. So that's what we mean about manually reviewed by expert scientists. The other thing about reviewed entries, the ones with the little gold star, they're also shown, this little icon here is shown in gold on the website as well. One of the things we do as a part of the process of curation is we group together isoforms derived from the same gene. So you have one entry which could have multiple isoforms for the proteins derived from that gene. And the unreviewed entries, which we start with, they've been through a process of computational analysis. And in this case, the isoforms are actually separate, so that there'll be multiple isoforms as multiple entries in this state when they're unreviewed in the tremble part of the database before they go through manual curation. Going back to our example, this is another example where instead of having gene names, I've got UniProt accession numbers or identifiers, and I could put those into the same search box and tell the website that I'm going to be using a UniProt accession, AC or ID, and then I want the gene names back. Another kind of search that one can do, moving on to a different aspect of the website, is this thing, a peptide search. So if you've got a peptide sequence derived from any protein of interest, you could type it into this dialog box and then run a peptide search. And in this case, I've chosen to restrict the taxonomy to human. And with this particular peptide, we get 3 hits, 2 which are for reviewed entries and 1 unreviewed. Moving on to a different way of looking at data in the website, I've chosen one particular example here of a protein that I've boxed in blue. This is the accession number, and then there's a name for the entry, IKKA human. This is its protein name. You can't see the full name, but if you were to click on these 3 little dots here in the parentheses, you'd find out its full name. And I think we'll see that actually in the next slide. And it's got other names as well. And you can customize this view here. You can add other columns to this view. And you can also change the way it's presented. This is called a table view. And now if we move on to, this is the entry. So its full name is inhibitor of nuclear factor kappa B kinase subunit alpha. Also, this is its gene symbol, CHUK, chuck. It's a reviewed entry. It's got this little gold icon, and it's from human. And on the left-hand side here, we've got highlighted in this purple box all the different types of data which are in this entry for this particular protein. And you can jump to any of them by clicking on the links here. And that allows you to explore different aspects of the data. Another way of viewing the entry, the information in the entry, across the top here, these things we normally refer to as tabs. There's the default view is you see the entry, but you can also see, for example, if this protein has known variants in it that have been curated, you can focus on those by clicking on the variant viewer. And then there's something called the feature viewer. This highlights different parts of the protein where they've been annotated. And another relatively new addition to the view of all the entries in certainly in some species, in human, I believe there may well be genomic coordinates in other species, possibly mouse, but I definitely know that human has them. So these are genomic coordinates. So what this means is that you can see the mapping of the exons that code for this protein onto the genome of origin. And slightly hidden here is also a link to publications for this particular entry, so where the data came from for some of the manually curated data. An example of the kind of information that we might have, and in this particular case, this is for this specific protein, is where it's an enzyme and we know what its catalytic activity is, we will have annotated that and then created links to other databases such as RIA. RIA is a reaction database, which is in fact where this image across the center of the slide comes from. And it shows that it's uses ATP to phosphorylate other proteins. And then as a result, it then produces these products, ADP and hydrogen ions. There will be links to the EC, the enzyme commission number, and will also indicate where this data comes from. So in this case, there are 3 publications which support this assertion. Another class of data that we capture by a mixture of manual and also automatic annotation is subcellular localization. And again, we've got here in this little icon with the bookmark in gold, this shows that this has been manually added. So the little gold bookmark means that a curator has added this to this particular part of the entry. And this is an interesting protein because it actually shuttles backwards and forth between the cytoplasm and the nucleus. I mentioned publications. This is an example of one of the publications that supports the assertions in this entry. You can choose to hide abstracts if you want to fit more onto a page, or you can view the whole thing. Then there are links to the full text where it's available in Europe PMC, and links to the journal as well. And this green box here, what this shows is how this publication was associated with this particular entry. So the ones with the gold icon here, that's been manually added by a curator. The little screen icon here with a tick in it, this has been added automatically as a part of the automatic annotation pipelines. There's a whole wealth of other data which is slightly hidden away in each entry, but if you scroll down, you'll find lots of different sources of information. So, for example, one of kinds of data that we capture, some of this is automatic. In fact, the majority of it is automatic, but some of it will have been added manually, and that is things like protein-protein interaction. So these are links to these databases that specialize in protein-protein interactions, but we have over 180 cross-references for other classes of data that link to other specialist databases. Moving on now from a specific entry and the kind of information you can find in that entry, a very obviously a key part of the information that we hold are the sequences. And so what I'm going to do now is say a little bit about how you can explore protein sequences and the features within them. And I'll start off by this is what you see at the bottom of each entry. But we can present the sequence in a whole range of different formats. You can download it, if the little icon here, where it says download. So you could choose FASTA if you wanted, or some other sequence format, and then you can choose to look at the sequence in a whole range of different ways. So this is something I mentioned earlier was the feature viewer. So left to the right across the screen here, we've got that protein I was talking about earlier, Chuck. So across the top here in gray is a sequence bar. I can't show you now without going to the live website, but you can choose to select the little gray grab bars at each end, and you can use them to zoom in on a particular region. So you could choose to highlight a particular part of the protein, and you can go right down to the individual amino acid level. Down the left-hand side here, we've got a whole list of different aspects of data for that particular protein. So does it go through post-translational modification? Are there specific domains known associated with the protein? Active sites, epitopes, so if there are antibodies or other molecules which recognize specific parts of this protein. Mutagenesis data, where this protein comes from a species which is amenable to mutagenesis. That kind of data will have been captured as well. And then whether a natural variants, we capture that automatically or manually. And proteomics data, the majority of this will have been captured automatically. And at the bottom, structural information. So on this particular screen, we've got a PDB structure, which is shown in green. This shows you that within an entry for this protein, the majority of the protein is represented in a structure, and there's a gap here where this particular region, either the structure is very poor quality or it was never included in the original data. And then further down, the AlphaFold data for this protein, and the different colors refer to the quality of the predictions from AlphaFold. And then perhaps something that's relatively recent, you may not have come across, is the alpha missense data. So this is now a prediction of the effect of changing an amino acid along the protein, what it might do to the structure. Zooming in on a particular part of this protein, if we're using the feature viewer, on the right-hand end near the C-terminus, in this particular protein, Chuck, there's a region which binds to a protein called NEMO. So not only can we select this by selecting the that particular region, this little brown box here, we can then, what that does is it highlights in a structure shown here in this window in the bottom right corner, it highlights the particular part of the structure in the 3D model. And in this case, it's the default is the PDB structure, which is at the top here with this identifier 3BRT. So these green, they look like there could be parts of an alpha helix. These green highlighted regions correspond to this region that I've highlighted in the this region of the protein which interacts with NEMO. Moving on from the initial sequence and the way it can be portrayed, let's say we want to find related proteins. So we can do a BLAST, and there are multiple ways you can run BLAST analyses from the website. Within each protein, there's a link to BLAST. So where it says tools here in this black box, in the toolbox, this allows one to choose a BLAST analysis, or you can look for IDs, related proteins. So let's say we run a BLAST. This would be the window we would see initially. So as I've said earlier, this is the protein we were talking about, the IKKA human, or Chuck. It'll automatically, when we go back to that previous slide, if I'd chosen BLAST here, this is what I would have seen. It would have automatically put the sequence into this box. And then one of the options here at the bottom is the target database. So the overall database is divided up in various ways. So SwissProt is the curated part of the database. And then I'm going to say a little bit about proteomes in a minute. Another very large part of the database consists of what we call reference proteomes. So together, this set is a large part of the database. And also I can choose to restrict my BLAST to a particular set of species, either one species or multiple ones. As an example of the kind of thing you'd see as a result, this is if I'd taken that previous sequence and run a BLAST, I would have got, the default is a maximum of 250 hits. So you may see 250 or you may see less, depending on exactly what your sequence is. In this example, I've just got 250 because that's the most the website will return. Excuse me. And then having got your BLAST, you can then filter it in various ways. You can choose to look at a particular set of hits which only come from one species, or have they got other kinds of, what you can't see in this display is I could scroll down here and there would be other options to filter the hits. So let's say I was only interested in some of the reviewed hits. So these are SwissProt entries which have been hit by our human sequence. So naturally it finds itself with 100% match, but it also detects a cow or bovine sequence and a mouse sequence, and they're very similar, as you can see from these percentages. As the default setting, you can see if I was to click on this hit, say the mouse sequence, you would see that, as I've said, they're very similar, and it'll show the alignment, and then you can choose how that alignment is portrayed on the screen with these little buttons here where it says overview and wrapped. So the wrapped one will show, you need to scroll down, and then you would see the other parts of the protein that are hit by your query sequence. Having got a few hits, one could then choose to do a multiple sequence alignment. And so if you've got 2 or more, you can then choose them from this tools box, or you can see there's a blue tick here, or a white tick on a blue background. We could do that manually. I could choose any number up to, I'm not sure what the maximum is. It might be 20. It probably is more than that. You can choose those and then create a multiple sequence alignment. I think it is more than 20. And then initially what one sees before the multiple sequence alignment is run is you see a screen like this. They appear in FASTA format, or if you've got a text file, you could choose to load your sequences from a text file. They would need to be in FASTA format. And in this case, there's a maximum of 50. And this is the kind of output that you would get having asked for a multiple sequence alignment. And what I've highlighted here in this blue box is some of the ways you can customize the way the multiple sequence alignment is shown on the screen. And so in this case, what I've chosen is I've used a human sequence as a query, and now I've got mouse, human, and cow sequences in the alignment. And then I can choose from the select annotation drop-down menu. I can say, well, I'm interested in knowing, for example, do these proteins all share a common sequence at the active site? So I could choose the active site from mouse and then ask the website to then highlight that residue or residues and then compare them across the three sequences or more. And in this case, you can see in the bottom right corner here, you've got a conserved glutamic acid, which is found in all 3 proteins, unsurprisingly, because they're very highly conserved. So, BLAST and multiple sequence alignment. I've given you a whistle-stop tour through looking at individual entries. I've talked about looking for identifiers for protein sequences and then searching for similar sequences and multiple sequence alignment. In this last couple of parts of the presentation, I'm going to talk about some of the other ways in which the data is presented. So I mentioned at the beginning, a proteome is all the proteins known to be expressed by an organism. And these will be derived from a sequenced genome. So this is one of the ways in which the UniProt data is presented. And we have over 160,000 proteomes. And this tree, kind of tree, tries to portray some indication of the richness of the data in UniProt, dividing up the different species belonging to the 4 superkingdoms, eukaryota, bacteria, archaea, and virus. So each proteome has a unique identifier, and then within that proteome, you can then navigate to individual proteins. Finally, the UniProt database, as well as the kind of information I've talked about already, has a lot of data about disease, and also in the case of model organisms, we have mutagenesis data, and I'll say a little bit about that in a minute. So if I was to look at the disease and variant section, which I've highlighted here for a human entry, this is what one might see. So in the example of a disease called Parkinson's disease, and it has its own acronym, PARC, there's a summary of the symptoms of the disease or condition, and then there'll be natural variants for which we have evidence that they have some association with that disease, and you can go into more detail if you were to look at a particular entry that these variants come from. In this case, there's actually a second variant of Parkinson's disease on our website called Parkinson's disease 2. I'm not going to go into any more detail about this now, but I'm going to move on to say a little bit about phenotypes which would, when you move from human into a model organism, so non-human, we talk about phenotypes rather than disease. And so in this example, this has been pulled from a fruit fly model of Parkinson's disease, actually. And in this case, I believe it's one of the proteins which is known to be involved in Parkinson's in humans has been mutated in fruit flies, and hence it gives rise to abnormal phenotypes. So, we'd describe those as disruption phenotype. And these would usually be either a null which has been created by deletion, for example, so a null mutant, or they may be knockdown phenotypes that have been created using, for example, RNAi or morpholinos. So morpholinos are short oligonucleotides that are used quite frequently in some model organisms to create abnormal phenotypes and knock down the expression of a particular gene. And the other thing that we display for mutations in these model organisms, we would give the mutant name. So, for example, this I believe might be, this is an example of a set of mutations for a protein from, in this case, from the worm, Caenorhabditis. So this mutation here at the bottom here, this fifth one down, this mutation is called M27. This particular mutant, rather, is called M27. And then we show the position of mutations, where they are in the protein, and then their effects with some kind of description. And again, this little gold bookmark here shows this comes from a publication and was manually curated. So I've given you a very rapid overview of how you can perform a search and look at the functional data in UniProt entries, how to analyze those sequences. Very briefly mentioned that we hold data in proteomes, so a set of related proteins all derived from one organism from their genome, and a little bit about disease and mutagenesis data. I mentioned early on that our data gets updated approximately every 8 weeks. It's all freely available, and you can download it in a whole range of formats: plain text, XML, other related formats, XML RDF, FASTA for sequences, and so on. Almost at the end here. Any feedback you can supply to us about the website is much appreciated. And I've highlighted here in gold some of the places you can go to send your feedback. So there's a little letter icon here. That's one way of sending feedback. And the other way is down here on the right-hand side of the homepage. There's a feedback. This is a more immediate feedback tool. What this does, it just gives you an icon, and you can choose to show how pleased or otherwise you were with the data for a particular entry. And I think it gives you 4 or 5 options about how pleased you were with the results. And if you want more detail, then you can click on the help in green here, and then at the top right corner, there's another help link. And you can contact us through the website. There's a dedicated help center, and there's also links to UniProt specific training. They will probably take you actually to the training website, and they are available in the help center. Excuse this this last image. It's slightly out of date because it talks about a release which is a few years ago. I mentioned the 3 different consortium partners at the beginning. This gives you an idea of the scale of the effort that's involved from those 3 different partners. Hopefully this is an up-to-date portrayal of all the people that create UniProt. Our money, our funding, the majority comes from the NIH, National Institutes of Health, but we also get money from support from EMBL itself, from Open Targets, and from SERI, which is a Swiss government organization which funds SIB, our partners in Geneva. Upcoming, at the beginning, Ajay mentioned that there are other webinars about UniProt services and data that will be coming along soon. One of them is this one, which describes how you can programmatically access UniProt using tools such as Python. And this one, I believe, might be coming up on the training website very soon. Thank you.
Great. Thank you, Paul. Thanks for the presentation today. And thanks everyone for joining in. But there's still many open questions in our Zoom Q&A, so I can read them out. So first one is, how does UniProt deal with incomplete or conflicting protein data from experimental results? And how can a user suggest correction to protein entries?
I can take this, Paul.
Okay.
So normally incomplete or conflicting sequence data that's published will be merged with an entry that represents that protein that's being researched. So we tend to try and merge everything that is from the same protein into a single entry if there's experimental evidence for it. If there's conflicting sequence data, we will include that as in a description as this is a potential conflict of sequence, and we will link to the reference that gives that sequence conflict. Regarding how you can suggest corrections or protein entries, at the top of each page on the website, there's a little envelope icon. If you click on that, you'll be taken to a form where you can enter any information that you want curators to look at. And this can be an update request, or if you find a typo or anything else that's wrong with the entry. If you enter it there, it'll be triaged and given to a curator to look at.
Great, thank you. Next one is it okay to choose the unreviewed proteins for research usage? If yes, why? And if not, why?
Definitely. They've been through a set of very well validated automatic annotation pipelines. We do a lot of due diligence on checking that there are minimal false positives, and that data is very reliable. There will always be some things that will need following up by that you always have to be aware exactly what the implications are of those annotations, but they are very high quality. So yes. Would you like to add anything to that, Dave?
Other than the only reason they're unreviewed is either that there's no experimental data out there. If people feel like an entry should be reviewed, if they find that there is experimental data, again, contact UniProt and we'll be happy to curate it and update it for you.
Yeah, that happens all the time. And yeah, the vast majority it is, as Dave says, it's simply because there's no literature for that particular protein.
Next one is more career-based. Does UniProt offer opportunities for remote internship or roles for researchers interested in contributing as scientists?
Well, one thing that we do already, and I didn't mention it, is that we do encourage users to submit their own, in a sense, their own annotations to papers. There's an aspect of the website, it's right on the homepage, but you have to scroll down to the bottom, and there is a link allowing you to suggest your own annotations for either for a paper from another group or from your own group, and that's a kind of dipping your toe in the water initial step. But beyond that, we don't have any curators who work entirely remotely. We're all involved in some way in the office some of the time.
All right. So next one is there an available tool for visualizing or mapping the surrounding of membrane-associated protein like the class GPCR? It would be nice to know components surrounding the protein in the membrane, such as which phospholipids are most likely to be near that receptor.
Not that I've come across, but I'm pretty sure future iterations of AlphaFold will be able to look at interactions with membranes, but I don't think we're there yet.
Okay, how can I see and create 3D structures of protein?
That sounds like something that is outside our, again, outside our immediate remit. You might be able to find people at PDB or, I suggest if with a question like that, our colleagues in PDBE might be able to help.
Right, thank you. Is there a way, I think it's again about programmatic access, is there a way to programmatically access or download all protein variant association from variant viewer?
Yes, is the short answer. You can do it that way, or so if you're talking about all the variants from a particular entry, that's quite straightforward from the variant viewer. But if you're talking about something larger scale, if you were interested in variants in a lot of proteins, you could do that through the API.
So, all right. So thanks, Paul, again for the presentation today, and thanks, Dave, for your help with questions and answers. Okay, and thanks everyone for joining.